50 million DNA profiles. Right now, sitting in private databases owned by two of the biggest consumer genetics companies in the world, there are 50 million genetic fingerprints. 50 million complete biological identities voluntarily uploaded by ordinary people who wanted to trace their ancestry and find distant relatives. And the FBI, the same agency with the legal authority to serve warrants on those companies and demand access to that data, has not used it to name a suspect in Nancy Guthrie's disappearance. The question this entire community has been asking for months is why. This week the most credible voice in forensic genetic genealogy answered that question publicly on the record. And the answer is not what most people expected. It is not a legal barrier. It is not a political one. It is not a resource problem or a processing delay or an investigative failure. It is a mathematics problem. And the software that solves it is being built right now, fast-tracked, specifically for cases like Nancy Guthrie's. Tonight we are going to explain exactly what that means, exactly what the obstacle is, exactly who is working to clear it, and exactly what happens to this investigation the moment they do. Hit that like button right now. Share this video immediately because Nancy Guthrie has still not been found and this development changes everything about the timeline of this case. Subscribe to Black Veil Crime Files with notifications on because when this technology delivers a result we will be the first channel breaking it down completely. And stay until the very last second because the timeline we are building tonight, from where the investigation stands right now to where it could be in a matter of weeks, is the clearest picture anyone outside federal law enforcement has put together on this case. Let us start with the technology itself. How genetic genealogy actually works, and why it is different from regular DNA testing. Most people who follow true crime cases are familiar with CODIS, the FBI's National DNA Database. When a forensic laboratory processes DNA evidence from a crime scene they generate what is called an STR profile. Short tandem repeat profile. That profile maps a specific set of genetic markers and uploads them into CODIS where the system searches for direct matches against profiles from convicted offenders and people arrested in jurisdictions that require DNA collection. CODIS is powerful. It has been responsible for millions of investigative connections since its creation in the 1990s. But it has one fundamental limitation that cannot be worked around. If the person responsible for a crime has never been in the criminal justice system, never arrested, never convicted, never had their DNA collected by law enforcement, CODIS has nothing to match against. That person is invisible. Completely. No matter how clean the DNA sample is. No matter how strong the evidence is. If they are not in the system they do not exist in it. Genetic genealogy solved that problem by changing the question entirely. Instead of asking whether crime scene DNA directly matches a known offender, genetic genealogy asks something different. Does this DNA match even partially anyone related to the person who left it? And because DNA is inherited, because you share portions of your genome with your parents, your siblings, your children, your cousins, your second cousins, even your third and fourth cousins, a database of ordinary people who share common ancestors with a suspect becomes a map that leads directly to them. But here is the critical detail that most coverage of this case has missed entirely. Genetic genealogy does not use STR profiles. It requires a completely different kind of genetic data called a SNP profile. SNP stands for single nucleotide polymorphism. A SNP profile is generated by analyzing hundreds of thousands of specific positions across a person's entire genome. It is the same type of profile that ancestry testing companies generate when you mail in your sample. It is what powers their ethnicity estimates and relative matching tools. To search genetic genealogy databases, investigators need a SNP profile generated from crime scene DNA. That profile gets uploaded to the database. The system finds partial matches with people who share enough genetic variants to suggest a family relationship. From those partial matches, a skilled genetic genealogist builds family trees, traces lines of descent, and eventually identifies the specific individual whose DNA was found at the scene. This framework works. It has been used to solve hundreds of cases. including the Golden State Killer, a monster who murdered at least 13 people, committed more than 50 sexual assaults, and terrorized 2 separate regions of California across 2 separate decades before being identified through genetic genealogy in 2018 after more than 40 years of evading capture. That case was considered permanently unsolvable. This technology solved it. Now bring that into 2026. Into Tucson. Into Nancy Guthrie's disappearance. Into an investigation with DNA evidence recovered from inside her home and the FBI actively involved. The pathway exists. The databases are accessible through legal process. The expertise is ready and standing by. So why has nothing happened yet? What Cece Moore said and why it matters. Cece Moore is not a peripheral figure in forensic science. She is in many ways the person who built this entire field. Moore is the chief genetic genealogist at Parabon Nanolabs, the forensic genomics company that pioneered genetic genealogy for law enforcement. By the time she spoke publicly about Nancy Guthrie's case, she had personally contributed to solving more than 300 criminal cases using this technology. 300. Including homicides, sexual assaults, and cold cases that had gone untouched for decades. She has testified in court as an expert witness. She has trained law enforcement agencies across the country. She has been involved in some of the most significant genetic genealogy cases in American history. When Cece Moore speaks about the capabilities and the limitations of this technology, she speaks from direct operational experience. Not theory. Not academic speculation. She has sat at the keyboard, uploaded the profiles, built the family trees, and personally watched cases break open that had been closed for 30 or 40 years. She is also not someone who speaks carelessly about active investigations. When she chose to speak publicly about the specific technical obstacles in Nancy Guthrie's case, naming those obstacles precisely, detailing the science, and naming the company working to solve it, those were deliberate choices. She said exactly what she intended to say. Here is what she confirmed on the record. The DNA evidence recovered from inside Nancy Guthrie's home exists as a complex mixture. Multiple contributors. Multiple people's genetic material physically mixed together in the same sample. And the technology to cleanly separate those contributions into an individual SNP profile that could be searched against genealogy databases does not currently exist in a validated, reliable, legally defensible form. That is the obstacle. And it deserves a complete explanation because it is the pivot point of this entire investigation. The complex mixture problem, why 50 million profiles cannot be searched yet. DNA mixture occurs when biological material from more than one person is deposited on the same surface and collected as a single sample. Think about every surface inside a home that multiple people touch, occupy, or inhabit over years. A kitchen counter. A door frame. A chair arm. Any biological trace from any person who has ever interacted with that surface, family members, visitors, contractors, caregivers, becomes part of whatever is collected when a forensic team swabs it. In a residential environment, a complex mixture is not unusual. It is often the rule rather than the exception. Forensic laboratories deal with DNA mixtures regularly. Two-person mixtures, a victim and a suspect for example, have established validated methods for analysis. If one contributor provided significantly more DNA than the other, scientists can often isolate the major contributor's profile cleanly. But when more than 2 people contribute to a mixture, or when the ratios between contributors are unclear, or when the DNA is degraded, the genetic signals from each individual overlap and interfere with each other in ways that current analysis tools struggle to separate. This is called a complex mixture. And complex mixtures are the point at which current forensic science, for the specific purpose of generating a SNP profile for genetic genealogy, runs into a wall. Here is why the wall exists at the SNP level specifically. For standard CODIS searches using STR profiles, scientists have developed mixture analysis tools that work reasonably well even with complicated samples. STR profiling examines a small number of genetic locations, typically 20 or fewer, which limits how badly the signals interfere with each other. But SNP profiling examines hundreds of thousands of genetic positions simultaneously. At that scale, the interference between multiple contributors' DNA becomes exponentially more difficult to resolve. An algorithm that successfully separates a two-person STR mixture may be completely inadequate for a three-person SNP mixture. The mathematics are fundamentally different at different scales. C.C. Moore confirmed the DNA from Nancy's home falls into this category. Multiple contributors. A complex mixture. And the software to cleanly separate those contributions into an individual SNP profile usable for genealogy database searching does not currently exist in a validated form. Without a clean single contributor SNP profile, the FBI cannot upload the evidence to ancestry databases. The system is not designed to accommodate a contaminated multi-contributor sample. The matches it would return would be statistically meaningless. And so the genetic genealogy pathway, which is potentially the fastest route from evidence to identity, is blocked by this specific technical gap. That is not investigators failing. That is the leading edge of forensic science encountering a problem that has not yet been solved at the scale required. Part 4, the company building the solution and the timeline that changes everything. C.C. Moore's public statement did not end with the obstacle. It ended with something else entirely. And this is the part that changes the entire picture. She confirmed that a company is actively developing software specifically engineered to solve the SNP mixture separation problem. Not generally. Specifically. A deconvolution algorithm capable of separating complex multi-contributor SNP mixtures into individual profiles at a level of accuracy and reliability that would be usable in active criminal investigations. She described this development as fast-tracked. That word matters. Fast-tracked does not mean expedited on a standard research timeline. It means the development schedule has been actively compressed. The resources, the personnel, the computational infrastructure, all of it accelerated for cases exactly like Nancy Guthrie's. Stop and hold that for a moment. Right now, as you watch this video, there is a company actively building the specific piece of software that stands between investigators and a potential named suspect in this case. This is not speculative development on a 5-year research horizon. C.C. Moore described it as fast-tracked. Present tense. Active. With a timeline being aggressively managed. And then she went further. She stated on the record that if and when investigators obtain a viable single-contributor SNP profile from the DNA evidence collected at Nancy's home, whether through this new software, through another separation pathway, or through the recovery of additional DNA evidence that is less complicated, she believes this case will be solved within weeks. Not months. Not years. Weeks. Possibly days. The FBI's Genetic Genealogy Unit is standing by. The databases containing 50 million profiles are ready and accessible through legal process. The warrant mechanism is established and functional. The family tree analysis that C.C. Moore and her team specialize in can begin immediately upon receipt of a clean profile. The pipeline from viable SNP profile to named individual is complete and operational. The only missing component is the profile itself. And the company that may produce the capability to generate that profile is building it right now. Why this community matters and what comes next. Let us be precise about what is confirmed and what is not. Because this channel operates on verified analysis, not wishful inference. What is confirmed on the record? DNA evidence was collected from inside Nancy Guthrie's home. That evidence exists as a complex mixture. Complex SNP mixture separation cannot currently be performed reliably with existing technology. Software to solve that specific problem is in active fast-track development. If a viable single-contributor SNP profile is obtained, C.C. Moore believes identification through genetic genealogy databases will follow within weeks. What is not confirmed? We do not know how many contributors are present in the DNA mixture from Nancy's home. We do not know whether investigators have already identified any of the mixture contributors through other means such as standard Cody's comparison using STR analysis. We do not know the specific timeline for the new software's completion or validation. We do not know whether other evidence such as camera footage, digital records, financial trails, or witness accounts has moved the investigation forward through entirely separate channels. What we can observe is this. The specificity of Cece Moore's public statement is itself informative. She did not speak in generalities about forensic science. She spoke about this case. She named the specific technical obstacle. She named the company working to address it. She named a timeline for what identification would look like once the obstacle is cleared. That level of detail in a public forum during an active federal investigation does not happen by accident. She could have confirmed involvement and said nothing more. Instead, she named the obstacle with precision and pointed directly at the solution being built. That framing, a solvable obstacle with a specific solution in active development, is meaningful when it comes from someone with her experience and credibility. It is not the framing of someone describing a dead end. It is the framing of someone describing a wall that is about to come down. There is also something worth saying about the 50 million profiles themselves. Genetic genealogy research has established that when approximately 3 to 5% of a population has uploaded their DNA to searchable databases, investigators can theoretically find a familial connection to almost any individual in that population. The United States has approximately 330 million people. 50 million profiles represent roughly 15% of the population. We are well past the threshold for near-universal coverage. If the person responsible for Nancy Guthrie's disappearance has any living relative who submitted their DNA to any major ancestry platform, there is a genetic path to their identity. The path exists. The technology to walk it is being built. Nancy Guthrie has been missing since February of 2026. She is 84 years old. She is a mother. She is someone whose absence has been felt not just by her immediate family but by the hundreds of thousands of people who have been following this case across platforms and across months. The sustained public attention this case has received is not background noise. It is a variable investigators account for. Sustained public pressure on missing persons cases produces better outcomes. That is documented across decades of cases. This community is part of that pressure. The genetic genealogy pathway is one thread. It is potentially the most powerful thread available if the technology gap can be closed. But it is not the only thread. The investigation continues through multiple avenues simultaneously. And we will be here tracking every one of them. If you have any information about Nancy Guthrie's disappearance, please contact the Pima County Sheriff's Department tip line or the FBI's Tucson field office directly. A reward of $1 million remains active. You can remain completely anonymous. Leave a comment below right now and tell me what you think about the timeline Cece Moore described. Weeks once the profile exists. Does that feel close to you, or does it feel like too many things still have to go right? Tell me what detail in the DNA science breakdown changed how you understand this investigation. And tell me what you think about 50 million profiles sitting in databases that investigators cannot yet use because the mathematics to separate one person's genetic signal from a mixed sample has not been solved. Subscribe to Black Veil Crime Files with notifications on. The technology is being built. The databases are waiting. The FBI's Genetic Genealogy Unit is standing by. And when this case breaks, we will be here with the complete breakdown. This is Black Veil Crime Files. I will see you in the next one. Blackheart.